Very much, Mary Salas, and can everyone hear me okay to start with? So great, thumbs up from the back, wonderful. So I'd like to, of course, start by thanking the organisers for the kind invitation to present at this meeting. I'd particularly like to thank Mary Salas and Matt, who've done a lot of the heavy lifting of putting this fantastic program together. And so today I wanted to focus on the different genotyping strategies that we've been using within the DEWAS program that Dave has already introduced, that we've been utilizing really in our ability to rapidly detect and respond to both wheat rust and wheat blast outbreaks. And I think we've already heard over this conference the amazing possibilities with genome sequencing and all the technological advances and also the applications and the advances there too. However, what's really important to me and both Dave as well is that all of these technological advances that have been made in genome sequencing are made available to the masses, so the widest possible demographic of our community. And this is where we started our MARPLE diagnostics project, which is focused around the wheat rusts. And the work I'm going to talk about now really is very much a collaboration between my lab at the John Innes Centre and Dave Hodson and his team at CIMIP, where we wanted to see how we could utilize the advances in genome sequencing and develop this into a kind of strategy which would be very simple for strain-level diagnostics for the wheat rust pathogens, and to make this strategy and this platform we were developing into something that could be utilized really anywhere, so particularly in resource-poor locations, or indeed in the back of Dave's car in Ethiopia, as shown here. And so when we started this project, we were really inspired, both myself and Dave, by the possibilities of this handheld MinION sequencer, this nanopore sequencer, and the fact that it had been used extensively to type individual strains of various viruses and bacteria, particularly in the Ebola outbreak in Africa. And so we really wanted to use this for the wheat rust and to use this for strain-level diagnostics. But when we started this program, we realized that wheat rust are a little bit more complicated than viruses, and this was going to be quite a challenge. And so, of course, as we've already heard, the wheat rust fungi, they have very, very large genomes. So if we compare the wheat rust fungi to, for instance, Ebola, the wheat rust fungal genome is over 10,000 times larger. And of course, the MinION sequencer, it's quite small, it's portable, but the one limitation is it generates a small amount of data compared to other sequencing strategies. And so this was going to be a challenge if we were going to multiplex samples. Also, of course, they're obligate biotrophs. And if we'd like to take a sample from the field, take a leaf or a stem and then sequence it, we're going to get a lot of the wheat genome too, and that's going to drown out any of the pathogen data because, of course, we're sequencing on a small portable device. And in itself, strain-level diagnostics, very, very complicated. So we needed a new strategy. We needed something that would generate small amounts of data per sample so we could multiplex, but also enrich for the pathogen DNA so we didn't get drowned out by the wheat genome, and use this for strain-level diagnostics, which was also going to be very complicated. So here we turned our attention in the lab to the potential of amplicon sequencing and targeting genes with high variation between individual strains. And so the reason we could utilize amplicon sequencing is because we'd already done quite a lot of studies doing genome sequencing of various isolates of our wheat rust pathogens. And I just want to focus now on the development that we've been doing more recently for wheat stem rust, where we'd sequenced a whole array of isolates of wheat stem rust from lots of different countries, over 24 different countries. And so we had a really good handle on the genetic diversity. And when we stared at this phylogeny, where we were able to define our individual strains, we realized that within this phylogeny, it's really only the sequences of the genes which are variable between strains, so vary in sequence between strains, that is really contributing to the topology of this phylogeny. Most genes have exactly the same sequence between strains. It's only the ones that are different that are really showing all the branches and the branch lengths in this phylogeny. And so we thought, well, we can just focus on those polymorphic genes. And there are around 1,600 polymorphic genes that are really making the topology of this phylogeny. So maybe we could do amplicon sequencing with 1,600 genes. Well, that's quite a large number of genes to do PCRs for and then sequence. And so we also wondered, well, maybe we could make this a bit simpler. Maybe we could take a subset of these genes. And so we did various analysis, and I won't go into all of the details today, to try to take different subsets at different cutoffs and then to compare the phylogenies that were created by those subsets using lots of statistical analysis, and then working out which of these phylogenies still looks exactly like the phylogeny that you make with all the gene sequences. And we homed in on around 392 genes, and these were sufficient to reconstruct a phylogeny that looks identical to when you do full genome sequencing. And from those 392 genes, we could successfully design really robust primers for 276 of those genes, and so that's what we went forward with. And of course, with the 276, we could reconstruct our phylogeny. It looks exactly like when you have all your polymorphic genes present. But we also knew if we were going to run this in the back of Dave's car, this was going to also be a bit challenging, and maybe not everything would work to plan, so maybe some of these genes might fail. So we wanted to make sure we had a good failure rate built in, so around 40% of the genes can fail in amplification, and again, you can still reconstruct your phylogeny with just a subset of 60% of these genes. And this is important because we want to make sure that the topology is maintained, even if we do this in challenging environments, and there are more challenges to the system, and some of the genes might fail in amplification. So now for wheat stem rust, we had our really nice set of genes that we could move forward with. The next step was to integrate this into the MARPLE diagnostics platform that we've been developing for a number of years, and was initially developed for studying wheat yellow rust. And to give a bit of an overview of how this system works. Firstly, you start by identifying the disease in the field. You collect an infected sample. We then have a very quick and dirty DNA extraction that's very, very easy to use. And then we do an amplification of our genes on a portable PCR machine. And when we amplify these genes, we pulled the primer sets into just 4 reactions. So you only do 4 PCRs for each sample. And then you get all your amplicons from your PCR, and then you can sequence those on the portable sequencing device from Oxford Nanopore. This then allows us to see the genetic code of those genes, and we can then analyze that using automated data analysis pipelines, and enabling us to define the exact strain that's present in that sample. And then that information can be integrated into the amazing early warning system that Dave's already so nicely explained. And I think what's great about this system is that, as Dave mentioned, you can go from getting your sample to knowing the strain in just 2 days anywhere. So you can run this also in any location. And it also isn't reliant on sending samples overseas for analysis. Everything is done by the sample collectors, ensuring that those people maintain complete ownership of the entire process. And also, when we were developing the system, we also thought it was really important to make sure that we could also monitor the fungicide target genes, so genes in the fungus that encode the proteins that are targeted by fungicides, to see whether or not there were any potential mutations that could be indicative of a decrease in sensitivity to various fungicides that are really key in controlling the wheat rust pathogens. And so to do this, we scanned the literature and we identified a whole array of different mutations that have already been reported in other pathosystems, so not for the wheat rust, but for other pathogens. And we built those into the system. And we also enabled in the system a kind of survey to be done. These genes are sequenced, and then any novel mutations that are present for the wheat rust fungi can also be identified. And so now when you run MARPLE diagnostics, you get an output, a report, and in that report it will assess whether or not there are any mutations in these genes that have been seen in other pathosystems. It will highlight those, and it will also look for novel mutations that might be unique to the wheat rusts. And this will help us to rapidly identify any potential changes in the sequence of those genes that could indicate a potential decrease in sensitivity, but more importantly, help us to fast-track those samples for further analysis in the lab to really see whether we are getting a decrease in sensitivity. And so, of course, when we finished our wheat stem rust platform, it was really important that it was trialed in the locations where we want it to be used the most. And so we've worked with partners, wonderful partners in Ethiopia and Kenya, and they trialed the system and they ran samples with this system. And what they found is that those samples that they processed belong to clades, which are known clades to be present in those regions. And so this means that the system is working really well. And of course, this makes Dave very, very happy. And so within the MARPLE diagnostics program, we also made sure that all the elements that were needed to use the system were contained within a single hard case. And so everything you need to run MARPLE diagnostics can be then moved between locations. It can go to the areas where it's needed most, creating a truly mobile and rapid response laboratory. And so right now we're supporting a number of MARPLE community hubs in lots of different locations in Kenya, Pakistan, Nepal, Ethiopia, Turkey, and just from 2 weeks ago now, Bhutan as well. So really fantastic to move there. And so these people are running MARPLE diagnostics routinely to type individual strains of our wheat rust pathogens. And it is an absolute privilege to work with all of these amazing teams around the world. But also when we were creating MARPLE diagnostics, of course, we were very passionate about this method being adopted by others. And we've heard already how it's already being used in South Africa. And it's being used in a number of different locations. And this is because everything that you need to run MARPLE diagnostics is freely available. It's all on our website. There's a training guide, the primer lists, the multiplexing system, the equipment list, and all the code needed to run the bioinformatics. And so you can download this and use this for anyone that's interested in running MARPLE diagnostics. And so through the DEWAS program, of course, we've had this great opportunity to roll out this MARPLE diagnostic system for the wheat rust pathogens. But DEWAS isn't just focused on the wheat rust, it's also focusing on wheat blast too. And we've already heard so eloquently already from Paula on the kind of spread of, sadly, wheat blast into Zambia and Bangladesh from its original locations in South America. And so I think this spread into Zambia and Bangladesh, and the fact that particularly in Zambia, we're seeing now on the border with Tanzania, we really need to have a good strategy in place to enable rapid monitoring of wheat blast so that we can keep ahead of this disease and make sure that we put in place all of those management decisions that are needed to keep this disease in check. And so within the DEWAS program, we also have taken kind of a three-step approach in order to respond to wheat blast and to integrate these advanced genotyping strategies into this program for diagnostics and surveillance, starting with the kind of lateral flow COVID-style tests that Dave has mentioned, which have been developed by collaborators in China and in Bangladesh. And these give us that yes-no answer. So this is really important in, for instance, Tanzania. If we were to identify any samples in Tanzania we want to check, see if it has moved over the border. If it hasn't, we can then do that through these lateral flow devices. But in areas where the disease is sadly already endemic, we also want to get a bit more information about the lineages that are present. And we can get that information through work that's been done by the Sainsbury Laboratory in Norwich in the UK, where they developed this system that they call T-Plex. And it's, again, an amplicon system, but relies on Illumina sequencing. And so samples are sent to them, and then they analyze just 84 SNPs, and that tells you the precise lineage of wheat blast that's present in that particular sample. But also at the Sainsbury Laboratory, they've gone one step further, and they're also doing full genome sequencing to give a really detailed account as to the particular strains that are present in the area. And so this is through a program called Get Genome. I highly recommend everybody should look this up. This is a program that was established by Sophie and Kamoon at the Sainsbury Laboratory. And this program is a not-for-profit organization that they established. It provides free genome sequencing for small genomes, so for bacteria and viruses mostly, for anyone who doesn't have access to those kind of facilities. And they also provide full training, helping people to go from sending their samples to the Sainsbury Laboratory, but then they will work with you to get out kind of like genome announcements and to help you with analysis as well. And so this is a really fantastic program that's been established at the Sainsbury Lab. And then through DEWAS, we get to make use of it because they're also helping us through DEWAS to do full genome sequencing on a number of strains of the wheat blast pathogen too. And so for the rapid tests, you've already heard that these are now kind of in advanced trials, and so we have seen that we are able to use these tests. They've been trialed in both Bangladesh and also in Zambia with Batasiba, shown here, and we were able to show that these tests do work. And as has been mentioned, it's really important to have these tests because Fusarium looks very much like wheat blast, and so people might be a bit confused just staring at the symptoms. I know I will not be able to determine the difference if I was sent to the field. And so these are really super helpful in distinguishing between Fusarium and wheat blast. And so it's really great that these are now being rolled out. And we hope to have these available in, for instance, East Africa, where we want to be really careful and keep good monitoring going. And then for T-Plex, the SNP-based genotyping assay, which analyzes just a small handful of SNPs through the analysis that has been done at the Sainsbury lab, looking at various samples that were taken from Zambia and also Bangladesh. What they've confirmed is this B71 lineage, which Paolo has introduced you. This was the pandemic colonial lineage that moved out of South America and into Zambia, and then also independently into Bangladesh as well. What they found through doing this SNP genotyping assay is that that one lineage is still present in those regions. And so there have been potentially no more new exotic incursions of new lineages into those regions. And so that just confirms that that single pandemic lineage is still present in Bangladesh and Zambia. But through the full genome sequencing, what they've managed to do is firstly also confirm that B71 is the only thing that's seen in Zambia and Bangladesh. But they've also started to see some diversification occurring, and that is only really detectable with the full genome sequencing. And so now we're seeing diversification of the pathogen lineages occurring within these countries, and so they're starting to change and adapt to these locations. And so that will be really interesting to keep a handle on as we go forward, as the Sainsbury lab continue to analyze more and more samples, both in Zambia and Bangladesh, but also helping others in the community to analyze samples from their countries too. Okay, so then in summary, I've shown you this new MARPLE diagnostic system, which we've been running for a few years now, working with Dave, and how this has been really great for doing kind of quick strain-level diagnostics at high resolution, and it's helping us to kind of get a handle on the wheat rust pathogens and the strain distributions, enabling us to advance kind of strain-level diagnostics in areas where they didn't have this capability previously. Also, we've heard already how important it is that we have a system for rapidly monitoring the emergent threat posed by the wheat blast pathogen worldwide, and how through DEWAS we're doing this through the development of these simple lateral flow tests to give the yes-no answers, right through to doing full genome sequencing and get that kind of much more detailed insight too, for monitoring spread and also diversification of our wheat blast pathogen. And so I'd just like to finish there by, of course, acknowledging everybody who has been working on this project. Particularly in my lab, I'd like to acknowledge Lysos, who is a phenomenal bioinformatician who's been working on the MARPLE diagnostics project. But I'd particularly like to acknowledge today, of course, Dave and Mary Sellis, who have really led us forward with the DEWAS program. It wouldn't be possible, none of this work would be possible without Dave and Mary Sellis keeping us all together and keeping us in check when needed. So thank you so much. And to all of our partners at the MARPLE hubs, thank you for also contributing to the project. And of course, our wheat blast partners, our funders. And thank you so much for your attention today.